Hello again biologists, Mr. Kinnear here with another video lesson for higher biology. We're on Unit 1, DNA and the Genome, and today we're going to look at cellular differentiation, which is Key Area 1.4. So this is where Key Area 1.4 sits in the unit. We've just finished Key Area 1.3 gene expression, and that is closely linked to the differentiation in multicellular organisms. So it's about the expression of genes in different cells, something I touched upon in those last video lessons. And you will know that your body contains a huge amount of specialised cells. So the 5 in front of you are all human cells. They have the same genetic information in each and every single one of them, but they're different shapes and they have different roles. And that comes about through the process of cellular differentiation. Now that's something we've talked about in S3 and National 5 biology, but not necessarily in the context of gene expression. So it's the process of expressing genes that are characteristic for that type of cell. And as you can see from this diagram, on the left hand side we're expressing neuron specific genes to create our neuron, and the right hand side expressing epithelial specific genes to create our epithelial cell. What's not shown in that diagram is that we are turning off the converse genes in those different cells. So on the left hand side we'll be switching off expression of the epithelial specific gene, and on the right hand side we'll be switching off the expression of the neuron specific gene. So it's turning on and turning off different genes that create our specialised cells. So we'll start the process by looking at animal cell differentiation. So as you can see on the left hand side we've got a very general animal cell, and then it can turn into a variety of different specialised cells. So we all know this story. It's the same for animals and humans. Fertilisation is the beginning. We've got 2 specialised cells, the sperm and the egg, that come together to form one cell, a zygote, and that zygote goes through the process of cell division, so 2 to 4 and so on and so forth. We do get to an early stage of embryonic life, which is the blastocyst, which, as you can hopefully see, is still just a bundle of unspecialised cells. There is no form of fetus as yet. When we do get to fetal stage, then as you can see we've got head, arms, legs, ears, eyes, and all of those things, and internal organs. So this stage we do have specialised cells. We have specialised cells, tissues, organs and systems already formed. So it's much the same as even an adult. So that's going to be important when we come back to this later on. So within the blastocyst and the adult we have stem cells. These are unspecialised cells found in multicellular organisms, and they can do 2 things. They can self-renew, which is copying to produce a new stem cell, and that will maintain the stem cell pool. So they're always self-renewing to produce new stem cells and keep that going. And the second thing they can do, which is what we're looking at today, is differentiation, which is specialising. They can become a specialised cell. That then goes on to replace dead or damaged cells throughout your life. So you and your body is probably only around about a year old in terms of brand new cells. Doesn't mean you're 1 year old, but the cells in your body are replaced on an annual basis at the very longest time. Some are replaced far more readily. So as we learned in National 5, we have 2 types of stem cells, embryonic and tissue stem cells. The first being embryonic, which are taken from the blastocyst that I mentioned earlier, a very early embryo, around about 15 days. And those cells are termed pluripotent. They can become any cell in the body, so they're completely unspecialised at this stage. No tissues, no form of a fetus, as you can see from this diagram. The second being tissue stem cells, sometimes referred to as adult stem cells, but that is a misleading term. They are found in many different tissue types within fetus, children and adults, although at one point it was thought that there were different types of stem cells, child and adult and fetal, but there isn't. These are multipotent. They can produce multiple types of cells within tissue. A good example being blood stem cells, which are found within the bone marrow. Those can be differentiated into the different blood cells, red blood cell, phagocytes, lymphocytes, etc. And as we finish off animal cell differentiation, just want to talk about some examples of gene expression within specialised cells. So a red blood cell, its job is to carry oxygen, you know that fine enough. It will express the hemoglobin gene, the protein that is involved in accepting oxygen. Create a shape in the cell, the biconcave disc, and destroy the nucleus, both of those involved in carrying more oxygen. Our cardiac muscle cells, their job is to continue to beat. They have extra mitochondria for ATP production, and their intercalated disc is helping in the coordination of muscle contraction. And lastly, nerve cell or neuron is to carry nerve impulses. They need to create these dendrite structures to allow for the electrical impulses to be received. The axons can be up to 1 meter long, so that requires extension. And again, the production of the myelin sheath to increase the impulse speed. These are things that are happening in terms of switching on genes within these specialised cells. We move on now to differentiation in plants, which has some similarities and differences. The differentiation occurs at the meristems, which are areas of unspecialised cells that can divide and differentiate. So again, just like our stem cells in animals. There are two places to find meristems: apical meristems, which are in the roots and the shoots, so growing up and down, and the lateral meristem, so that's between xylem and phloem in a plant stem. That actually results in the annual growth ring, so that is how a plant will grow out the way. Within our growing shoots, we have different zones. At the tip, we have the zone of cell division and mitosis, so we're dividing cells and adding more cells in. Below that, the zone of elongation and vacuolation, which increases the length of our cells. Then they become differentiated, so specialising to things like our xylem and our phloem, and then they become permanent tissues below that. So we're always replacing at the tip and then elongating, then differentiating, and then below that becoming permanent tissues. And again, to finish off plant cell differentiation, let's talk about the gene expression that's happening in some of these specialised cells. So in the phloem cell, which job is to carry sugar around the plant, they need to form the companion cell, perforation of the cell walls, and the sieve tube contents need to die. In the xylem, which carries water up the plant, we need to deposit lignin in those rings or spirals that give it strength. We need to connect the end walls and destroy them to provide this ongoing tube. And in the palisade mesophyll cells, which is the major site of photosynthesis, they need to produce lots of chloroplasts and the pigment chlorophyll. Also as well, they need to produce the photosynthetic enzymes, which again is down to gene expression. Again, their cell wall shape to allow for close packing towards the top of the leaf for photosynthesis. So to summarise the key learnings here, in specialised cells, the genes required to produce proteins for that type of cell are expressed, i.e. switched on. All the others are switched off. Our embryonic stem cells, we need to know that they are in early life only. They come from a blastocyst and they are referred to as pluripotent. They can become any type of cell. Tissue stem cells are throughout life. We have them now, and they are referred to as multipotent. They can become a variety of cells, but not every single cell. And in meristems within plants, they are available throughout life and will be pluripotent. Again, they can become any type of cell within the plant. Stem cells are of great interest in many forms of therapy, including skin grafts, where we can grow new skin in a lab for burn victims, bone marrow transplants, so the blood stem cells found in bone marrow to treat leukemia, cancer of the blood, or other blood disorders, and even corneal stem cells, so we can culture and transplant these to treat corneal damage. It's also of great interest in research. It does require a stem cell line to be formed, where we culture stem cells and grow them within the lab. But it does help us understand growth, development, differentiation itself, and how diseases develop. It's also seen as an alternative to animal testing, so we can culture animal tissue without having to harm any animals. And it's seen that there could be potential treatments for Alzheimer's, Parkinson's, Duchenne muscular dystrophy, and spinal injuries, to name but a few. But that does come with some ethical issues. There's concerns over the capability and how far people could go with it. But these things are regulated in the UK by the Human Fertilisation and Tissue Authority. There's religious concerns, so people believing that life begins at fertilisation, and they want blastocysts to be treated as a living being would. An alternative around that is this new technique of induced pluripotent stem cells that avoids using embryonic stem cells. So what they can do is take stem cells from an adult, so tissue stem cells, and reverse the process and convert them back into their embryonic stem cell form, which means they are of far more use, or the same use, as the embryonic stem cells. That then circumnavigates the problem that people have with the blastocysts being used and destroyed within these therapies and research processes. And that brings us to the end of today's lesson. We've talked about the functions and characteristics of meristems, embryonic and tissue stem cells. I also mentioned that stem cells are incredibly useful for research and therapeutic use, but there are some ethical concerns when it comes to the use of stem cells, which means they require regulation. I would say it's worthwhile for you to understand those ethical concerns, as well as having a couple of examples of the uses of stem cells within therapy and research. But as always, if you have any problems, questions or queries, get in touch and I'll speak to you soon. Thanks, bye.